Today we are going to discuss about Mendelian inheritance. So monogenic disorders. What are monogenic disorder? Monogenic disorder is an inherited disorder which is caused due to defect in a single specific gene. Monogenic disorder is a type of disorder which will take in a single gene and that gene can produce a large effect. For example, some genes are defective but they don't produce much effect, but some genes, if they are slightly defective, they can produce a large effect. So therefore genes which are responsible for large effects, if they become mutant, it will produce a disease, and we say such disease as monogenic disorder. The inheritance pattern in monogenic disorder is of two types. So we have typical or classical pattern or Mendelian pattern, and we have atypical or non-classical Mendelian pattern. So classical Mendelian pattern is divided into autosomal and sex-linked. Autosomal can be dominant and recessive. Sex-linked can be X-linked and Y-linked, and X-linked is further divided into dominant and recessive. Non-classical Mendelian pattern of inheritance or atypical inheritance pattern example is mitochondrial inheritance pattern, trinucleotide repeat expansion or trinucleotide repeat mutation, and imprinting. In non-classical Mendelian pattern of inheritance, all the other patterns of inheritance which were not following the Mendelian laws were put together, and we have seen their examples already as trinucleotide repeat, mitochondrial inheritance pattern, and imprinting. Most of the monogenic disorders follow the principles of George Mendel, and so they are classified under classical Mendelian pattern of inheritance, which can be autosomal, sex-linked, like I had already mentioned. In autosomal monogenic disorder, the mutant gene is present on autosome, that is chromosome 1 to 22. In sex-linked monogenic disorder, there is a problem with either the X or the Y gene. The X gene is mutant or the Y gene is mutant. X-linked, what we call monogenic disorder, are more common than Y-linked monogenic disorders, which are few and are rare. So sex-linked monogenic disorders are those disorders where gene present on the X or Y chromosome is mutant and produces a disease. Now, before we go ahead, we need to clear some concepts such as what is genes. So, gene is a sequence of particular sequence of nucleotide or DNA, which is associated with formation of a particular functional RNA. With that RNA can be rRNA, mRNA, tRNA, or snRNA. A single chromosome has a single double-stranded DNA, and that double-stranded DNA has many genes, and there are genes have nucleotide sequences, and those nucleotide sequences are responsible for formation of different functional RNA. What are alleles? Alleles are different variations of a gene which are present at a specific position on homologous chromosomes. Locus is a specific position on a homologous chromosome. One homologous chromosome has many loci, and homologous chromosomes are similar chromosomes which are inherited from mother and father and have similar set of genes or alleles. Example of homologous chromosome is chromosome number 7, that is paternal chromosome number 7, and maternal chromosomes number 7 are said to be homologous chromosomes. So here if we take example, this is the maternal chromosome number 7 and this is the paternal chromosome number 7, so they are said to be homologous chromosomes, and these black dots on these what we call DNA or genetic material of the chromosome represent loci. So this loci is responsible for eye color, this loci is responsible for nose, this is responsible for mRNA, tRNA, sRNA. Similarly, if we have a loci on the same, if we have what we call, if we have a gene, so this is the gene for eye color, this is gene for nose, and mRNA, tRNA, sRNA. So if we have a gene on the same loci of the homologous chromosome, then it is called as allele. So this is allele of this is allele of this, because they both are going to what we call manifest the genes for nose, formation of nose. So these genes which are present on homologous chromosomes at specific loci or position, these genes have a special relation with each other and are called as alleles of each other. Different variation of genes or alleles are present on homologous chromosomes on the same loci. One important point to remember here, maternal 7 chromosome and paternal 7 chromosome are said to be homologous, but maternal 7 chromosome and paternal 8th chromosome are not homologous chromosomes. So the number should be same. Then now we are going to see what is genotype. So genotype is basically the genetic makeup of an organism, and it is the information present on the genetic material of the organism. Phenotype is the observable characteristics produced by the expression of genes, and phenotype depends on the genetic makeup or genotype and environmental factors. And now we are going to understand the concept of dominance and recessiveness. So now we are going to understand the concept of dominance and recessiveness. To understand the concept of dominance and recessive, we are going to consider that these are two homologous chromosomes. Chromosome number 1 maternal, chromosome number 1 paternal. And on this here we have two genes on top, and these two genes will consider as brotherly genes, and here we have two genes on the bottom which will be considered as sisterly genes. So the basic concept here is that brothers usually, if one brother won't do work, he will not let other brother also do work. That is the relation between brothers. And the sisterly genes, the concept sister relation is that if one sister is sick and she can't work, the other sister is going to help and do double work and make up for the work that is left behind by the other sister. But in the case of brother, that's not the thing. If one brother doesn't work, he will spoil the work of other brother also. So now what happens here is, if we consider that this maternal gene produces mRNA which produces a peptide, and the paternal gene produces a peptide, mRNA which produces peptide, and both of them together form a normal structure. So if one of the brother gene gets mutated, the normal gene will produce the mRNA, it will produce its peptide, and the abnormal gene will also produce abnormal mRNA and the abnormal peptide. But now to form the what we call respective peptide, the normal peptide and the abnormal peptide will react with each other, but the abnormal or the mutant product will disrupt the healthy product also. And in spite of the healthy product present, there is some structural problem because the abnormal protein is manifesting its effect even on the normal product. So this was brotherly gene effect. So since there is a structural protein problem, the person is going to develop observable abnormal phenotype. So very important part, that abnormal phenotype will be manifested by the mutant allele in the presence of healthy allele. It means that in the presence of healthy allele, the mutant allele can produce abnormal phenotype. This means that mutant allele is dominating over the healthy allele. So the phenomenon of brotherly gene is that healthy allele plus mutant allele, it is going to be dominant and eventually will produce the abnormal structure. Another easy way to remember this is that the story of two brothers that their parents told them to cut the grass of the lawn before the guests come, but one of the brother didn't want to do the work, so he started disturbing the other brother also, and both of them did not cut the grass of the lawn even when the guests came. Now we will see the concept of the sisterly genes. So we remember the concept of sisterly gene is that, as an example, that if the sisters were given the work to cook for the guest, if one of the sister gets sick or mutated, the other sister will do double work and compensate for this sister. So here the normal allele of the gene is making the enzyme, but the defective or mutant what we call allele of the gene is not making any enzyme, so it will not disrupt the product which is being formed, and the product being formed will be normal. Here even feedback mechanism can work. For example, if A plus B substrate forms C plus D and A plus B substrate increases, it will put a signal to the normal allele of the gene to produce more enzyme to balance the equation. So feedback mechanism is also possible in sisterly gene mechanism. So this means that in sisterly mechanism, mutant allele of the gene in the presence of healthy allele of the gene cannot produce abnormal phenotype, and it is recessive in nature, unlike the brotherly gene in which mutant allele could produce abnormal phenotype even in the presence of healthy allele of the gene, and it was dominating. Therefore, genes can behave in two ways, either brotherly way or sisterly way. In brotherly way, dominant behavior is observed, and sisterly recessive behavior is observed. Dominant behavior is usually associated with genes which are responsible for producing structural and regulatory proteins, whereas recessive behavior is observed mostly in genes which are responsible for producing functional proteins such as enzymes. Regulatory proteins are receptors. So the conclusion of this brotherly and sisterly gene is that it means when there is a significant mutation in genes which are concerned with the structural proteins, even if one allele is defective, abnormal phenotype will be there. So as we saw here, these brotherly genes are mostly associated with structural genes and regulatory genes. So even if one gene is mutant, the phenotype will be affected. But when significant mutation in one allele of enzyme related genes is what we call present, the abnormal phenotype will not be present. So as we saw here, in sisterly genes or are the genes which are associated mainly with genes producing functional proteins or enzymes. So if there is even one mutant gene, it will not affect the activity of the other sister gene, and the phenotype won't be abnormal. Now we are going to see the concept of homozygous genotype, heterozygous genotype, homozygous mutant allele genotype, and allelic heterogeneity genotype. So if two alleles on same locus of a homologous chromosome are exactly similar to each other, they are called as homozygous genotype. So example, this is what we call, these are two alleles which are on the same locus of homologous chromosomes. So chromosome numbers maybe 5, chromosome number 5, paternal, maternal, and they're exactly similar. See, now changes, they are called as homozygous genotype. Homozygous normal allele genotype. If two alleles on same locus of homologous chromosomes are somewhat different, they are called heterozygous genotype. So here they are almost same except for the I part, and they are on homologous chromosome on the same locus, then they will be called as heterozygous genotype. Then we have what we call, if two alleles on same locus on homologous chromosome are same but they have mutation, okay, but they are both mutant. So this is not normal. Normal was this, but these are not normal. They are mutant but they have the same type of mutation at the same locus at the same place in the gene, then it is called as homozygous of mutant allele genotype. Then if we have two alleles on same locus on homologous chromosome, but they both are mutant at different places of the gene, okay, in this gene the mouth is mutated, in this allele the eye is muted, then this is called as compound heterozygosity or allelic heterogeneity. In allelic heterogeneity, both alleles are mutant, but they are mutant in a different way. Therefore, the mutant product are slightly different from each other. This is also called as compound heterogeneity. So, in allelic heterogeneity, mutation is of same alleles, but the type of mutation is different. Therefore, they produce different products and manifest disease differently. Example of this can be sickle cell anemia and thalassemia. So further carrying this concept of allelic heterogeneity, it is possible that the mutation which occurred on your chromosome number maybe 15 locus may be different from the mutation that occurred in chromosome number 15 of Dr. Najib. So therefore different mutation leads to slightly different clinical manifestation. So here we have two examples. Here one nucleotide was altered, so the what we call missense mutation took place, and the missense mutant product one amino acid different from the normal peptide. Okay, so here's the length of the peptide. It was somewhat similar, but still it was mutant. In this, what we call allele, there was mutation which took place in such a way it formed nonsense mutation, and there was premature what we call appearance of stop codon, and that resulted in formation of truncated protein due to premature translation. So this proves that different mutation can have different manifestation according to allelic heterogeneity. So example of allelic heterogeneity is that sickle cell anemia and beta thalassemia, like I told. In sickle cell disease, mutation in gene is because in the gene associated with beta globulin, we know hemoglobin beta alpha chain and beta chain. So beta chain is affected and beta chain is normal. Here at 6th position, the amino acid glutamic is replaced by valine, and due to that we have abnormal beta chain. In some other person, the same gene may be altered in such a way that the mutation produces an early stop codon. So in this, the beta chain produced is partial and undergoes destruction early due to being an abnormal protein. So this person RBC beta chain will be less, and he will have beta thalassemia due to low number of beta chains. So here again we saw that mutation in the same gene, but different type of mutation was there, which led to different structural, functional and regulatory protein manifestation. And so the manifestation disease is also different, and this is an example of allelic heterogeneity. So now just to recap it, most of the patients having autosomal dominant disease are usually heterozygous. That means they have one defective gene and one healthy gene, or one defective allele and healthy allele. And most of autosomal recessive disorder patients will have both the alleles which are defective or abnormal. That is, they can be homozygous of mutant allele. So here we can see that the recessive will be like this, homozygous or mutant, or they can be compound heterogeneity for recessive disorders. For dominant disorders, it will be like this heterozygous situation where one is defective and other is normal allele. A recessive trait or gene is said to be present when in the presence of the healthy allele, the defective allele cannot make abnormal phenotype. Whereas a dominant trait or gene is said to be present when in the presence of healthy allele, the defective allele makes abnormal phenotype. Mutation in structural gene causes mostly dominant disorders, and mutation in enzymatic genes causes recessive disorder. Now we have a question that as a group which is more severe, autosomal dominant diseases or autosomal recessive diseases. So the answer is that autosomal recessive diseases are more serious and dangerous because both the copies of the allele are defective. But in autosomal dominant, the is relatively less serious as compared to autosomal recessive, because at least one of the gene is healthy and the other allele is defective. Another concept is that when genes which are recessive in nature, they will have loss of function, whereas genes which are dominant in nature will have gain of function. So whenever there is gain of function mutation, that means it is a dominant mutation. So for example, if we consider these are two chromosomes and these two genes, this gene and this gene is responsible for formation of a particular receptor. So in gain of function, what happens that even though the stimulus or the ligand to this receptor is not present, this receptor, since it is defective, since the gene is defective, it will send abnormal intracellular signals without even the presence of stimulus, and that is an example of gain of function. Another example of gain of function, we remember that since one allele is defective out of both of the alleles, and this is brotherly relation, the structure of the protein which is going to be formed is also going to be abnormal due to the dominant nature of the abnormal protein formed by the abnormal gene, whereas there will be suppression of the a product of the normal gene, and so as a result this full protein will also be abnormal, and that is also called as gain of function. Loss of function mutations are usually associated with autosomal recessive disorders, and autosomal recessive disorders are mostly associated with enzymes. So in loss of function, mainly the problem is that either the enzyme is not produced at all, or the enzyme which is produced is in less amount, or the enzyme is produced in normal amount but the enzyme being produced is defective. So there will be loss of function. Now we have a question that as a group, which disease appears early or which has an earlier onset of disease, autosomal dominant or autosomal recessive? So the answer is that autosomal recessive diseases are present at earlier age or manifest their effects at an earlier age because both the genes are defective or mutated in the person, and when the person is born he will have no enzyme, so there will be severe disruption of the metabolic pathway. As a result, the disease will manifest very early. But in autosomal dominant diseases, since the patient has one defective allele and one healthy allele, the healthy allele can somewhat what we call produce some healthy products, and the disease will appear later in the life. Now, as a quick recap in autosomal dominant versus autosomal recessive diseases. So, autosomal dominant diseases are the diseases which are related in mutation in structural genes or in regulatory genes, whereas autosomal recessive disorders are the disease which are related to mutations in enzymatic genes. Then dominant disease manifest when the person is heterozygous, and recessive disorders manifest when the person is homozygous for mutant alleles. So this is the example of autosomal dominant where there is heterozygous, and what we call in recessive it will be homozygous of mutant allele, or it can also be allelic heterogeneity. I'm sorry, I'm not sure whether it will be only homozygous. What we call recessive disorder will only be homozygous for mutant alleles, or it will be homozygous for mutant allele as well as allelic heterogeneity. So I need to confirm that point. The next point of difference between dominant and recessive disease is that as a group, dominant diseases are less severe as compared to recessive disorders, and also the manifestation of dominant disease and their diagnosis are at a later stage of age, whereas recessive disorders are manifested earlier and are diagnosed also earlier in the life of the person. And dominant disease are mostly gain of function mutation, whereas recessive disorders are mostly loss of function mutation. Now we are going to study in detail about the autosomal dominant and autosomal recessive diseases and etc. about the pedigree charts. So first we are going to discuss Marfan syndrome. So Marfan syndrome is a disease which is caused due to mutation in the chromosome number 15, and this results in that the fibrillin production with the fibrillin produced is not acting as normal scaffolding protein for elastic tissue. So there is a problem with the fibrillin protein, because fibrillin acts as a scaffolding protein for elastic tissue or elastin. So the result of this Marfan syndrome, we will have three main groups of what we call problems. The first is ocular abnormalities, that is problem with the eyes, CVS abnormalities, and skeletal abnormalities. So ocular abnormality means that the suspensory ligament or the zonules which hold the lens in position, their elastic tissue is not very strong and the lens cannot be held properly in position. So here we can see these are our suspensory ligaments. So if they are in Marfan syndrome, see, they will not be very strong enough and they cannot allow the lens to what we call function properly, and the lens will bulge. So they will have ocular abnormalities. Then they are going to have skeletal abnormalities, that is, they are tall, they have long fingers, and then they have serious abnormalities such as that the elastic connective tissue in aorta and valves are abnormal. As a result, if the elastic connective tissue in aorta is abnormal, there can be what we call in the tunica media layer of aorta, elastic fibers can undergo degeneration and the dissection of aorta can occur. As a result, we know the blood should go through the lumen of the aorta, and here what will happen, the blood will start going through the between the lumen and between the layers of the aorta, and that can cause dissection of aorta. And if the elastic connective tissue in the valves are abnormal, then there can be problem with the valves of the heart, and that can lead to regurgitation. So this was the three main problems: Marfan syndrome, ocular abnormalities, skeletal abnormalities, and serious abnormalities. Now we are going to observe the autosomal dominant disorder of Marfan syndrome in a pedigree chart. So here we have a normal male and an affected female who has Marfan syndrome. So we are going to see their pedigree chart. What we are going to do, we are going to take this step separately, this step separately, and this step separately, and discuss them in detail. So let us start the discussion with the marriage of a normal male and a Marfan syndrome affected female, and let's see what is the problems which their progeny is going to face. So since the male is normal, the sperms will have either two of the chromosomes during gametogenesis, and the sperms will are all healthy because they have the normal what we call alleles, homologous chromosome. Now the female, when she makes the ovum, she will either put the healthy alleles in one ovum and the abnormal alleles in the other ovum. So there's 50% chance of the ovum carrying defective alleles and 50% chance of the ovum carrying healthy alleles. This is according to law of segregation. So now if we draw the pedigree chart for this, what we come to know is that the first child is going to get the first from father, first from the mother. So he's going to be normal phenotype and normal genotype because he doesn't have the abnormal allele. Whereas in this child, since he's heterozygous, so he will have the Marfan's syndrome or any other syndrome of autosomal disease, which we are going to discuss. And then the third child is also having both the normal allele, so he's going to be normal. Whereas the fourth kid is again having heterozygous manifestation, so as a result, he will be expressing the disease. So in autosomal dominant disorders, if there is mating of a normal person and a disease carrying person, the result will be what we call half mutant gametes of will can go of this couple can go to either of the kids. So there's a 50-50% chance to produce normal baby and an abnormal baby. Now we had seen the first generation, we saw that the male was normal, the female was abnormal, and then they had 1 2 3 4 kids. Now we are going to take the second case where in the second generation the affected male is going to marry a normal male. Let us see what happens in his progeny. The thing is that this case is also similar like this case. How here the male was normal and the female was abnormal, and in this case here the male is abnormal and the female is normal. So the chance of their progeny having the disease is going to be 50-50%, like in the first case. So we discussed that the chance of their kids having the Marfan syndrome is going to be 50-50%. What if this couple decides to have another kid? What is the chances of that kid having the disease or the Marfan syndrome? So the answer is that the chance of that kid having the disease is also 50-50% because the zygote formation is independent and is regardless of the previous pregnancy, because each what we call pregnancy is an individual event, and the chance of having the disorder can be 50-50% because the separate chromosomes can come and depends on the whatever the kid gets in his genotype. So therefore in autosomal dominant diseases, chances of normal and abnormal kid in every pregnancy is going to be 50-50%, and males and females are affected almost equally in number. Usually in autosomal dominant, proband has a history of affected parents. So what is proband? So here this and this will be considered as proband, that is the affected individuals who are under consideration. So in and they have an autosomal dominant disorder, so such people are called as proband and they have a history of affected parents. So here this boy and this girl had an affected father. Now we will discuss the pedigree of the first kid of this couple and see what is the result of its progeny, genotypic or phenotypic result of its progeny. The thing to remember here is that this person is normal, but we should remember that her mother was affected. So now what will happen in her kids? What is the probability of the disease being manifested? So for this kid, we don't know the genotype because she did not manifest Marfan's syndrome, but we remember that her mother or parent had the defective genotype. But when she gets kid, one of her kid is affected with Marfan syndrome. Why is that so? This is a very rare condition in which the mother was carrying the abnormal allele, but the mother herself did not manifest the disease, and this is a very rare case in autosomal dominant diseases. So this is a very unusual thing that the woman was having the autosomal dominant allele in her genotype, but she did not what we call manifest the disease, but eventually her kid had the disease. So why is this so? Here we come with introduction of the phenomena known as penetrance. So what is penetrance? To understand penetrance, we can go along with this example, that is that there was no abnormal phenotype, and this could be like because there was influence of environmental factors such as diet or exercise or influence of other gene, and those environmental factors and influence of other gene reduce the penetrance of the defective gene. This means that the disease was present up to the genetic level or in the genotype, but it could not penetrate deep enough to go up to the level of observable physical manifestation or phenotype. So this means that the defective gene was present, but there was no clinical manifestation or there was no phenotype. So such patient, we will say that, for example, this was a female, we will say that the female is expressing reduced penetrance or the patient is a non-penetrant patient. This phenomenon is not very clear and scientists are still researching on reduced penetrance. So this woman who had an affected mother and herself she was normal, but her daughter was manifested with the disease. This is because the woman was carrying the defective allele, but the penetrance of the allele in her was very low, and that is due to reduced penetrance, whereas that could be due to her environmental factors, etc., or the influence of the other gene. To understand reduced penetrance better, we are going to take the example of LDL receptor and its chromosome. So LDL receptor expressed on the hepatocytes, and from blood circulation, LDL, which is rich in cholesterol, captures by receptor and is internalized in hepatocyte. So this means that LDL receptors are expressed on hepatocytes, and in blood the LDL which is circulating that will carry cholesterol with it, and LDL receptors will recognize these LDL in blood and will capture that LDL and enter into hepatocytes. So basically LDL receptors help the liver to understand the level of cholesterol in the blood. So if cholesterol level is low, as a result hepatocytes will produce more cholesterol to balance it out in the blood, and if the cholesterol level is high in the blood, the hepatocytes will what we call reduce the internal production of cholesterol in the liver and balance out the level of cholesterol in blood. So LDL receptors help in regulation of the cholesterol level. Now what if this LDL receptor is mutant? That means either the LDL receptor is not present, or the LDL receptor is present but it does not take the LDL or cholesterol inside the cell of hepatocytes. So as a result, the hepatocytes will become blind to the level of circulating cholesterol in the blood. Hepatocytes can assume that there is no cholesterol in the blood because there is no cholesterol or LDL coming into the hepatocytes. So what they do, they tell the liver to produce more cholesterol, and as a result the blood will be flooded with cholesterol, and due to this the patient will develop hypercholesterolemia, and this is also known as familial hypercholesterolemia because it is what we call genetic disease which is inherited, and it is due to mutation in the what we call gene which is responsible for production of LDL receptor. Now let us imagine that this is the pedigree chart of a normal person marrying a person with familial hypercholesterolemia. So this means that there is a 50% chance that his offspring will be normal and 50% chance that his offspring will be abnormal. But what if this woman who is carrying the familial hypercholesterolemia defective disease gene, and what she does is that she consumes very low cholesterol and does good exercise? It can be possible that the disease of hypercholesterolemia is not manifested in her because she doesn't eat much cholesterol and she does regular diet. So in spite of the abnormal genotype, the environmental factors such as diet and exercise will modify the phenotype. So as a result, phenotype depends on the genotype as well as the environmental factors. The woman herself may not develop the manifestation due to a good diet and healthy lifestyle of doing exercise and all, but her children might develop because the children might not be so health conscious and they might be eating so much junk food, so their cholesterol level already be high and they might not follow or do much exercises, so they can what we call develop familial hypercholesterolemia, and this can also be stated as an example of what we call reduced penetrance because the woman herself did not show any manifestation of familial hypercholesterolemia because she was being helped by the environmental factors such as low diet consumption of cholesterol and good exercise, but the disease appeared in her children because they were not so health conscious. So just to recap, penetrance or reduced penetrance, abnormal genotypes is present in the person, but the person does not develop the abnormal phenotype. Such cases are called as reduced penetrance, and that reduced penetrance can be due to environmental factors. Example, 100 people with abnormal genotype will express, if 100 people they have the abnormal genotype for Marfan syndrome, 70% of those people will manifest the disease. So that means penetrance is 70%, whereas 30% of them will have no manifestation. We can say that penetrance. We have understood the concept of what is reduced penetrance, and we had seen different what we call pedigree charts. Now we have to understand two more concepts, rather three more concepts, that is pleiotropy, variable expressibility, and new mutation. So what is pleiotropy? Pleiotropy is the phenomenon when there is defect in one gene, and the defect in one gene can produce clinical manifestation in multiple systems. So what does this mean? Example, in Marfan syndrome, we remember the mutation is in chromosome 15, and it is in the gene which is associated with production of fibrillin. As a result of mutation in this gene, there will be clinical manifestation in multiple systems such as ocular abnormalities, skeletal abnormalities, and CVS abnormalities. So there was defect in one gene, but that led to the problems in three different what we call systems. So this is an example of pleiotropy. Another example of pleiotropy is osteogenesis imperfecta. In osteogenesis imperfecta, the collagen type 1, there is defect, and due to this, there will be defect in the bone formation. The person will have blue sclera, and the blue sclera is due to thinning of the collagen near in the eye, and then there will be venous plexus is seen, and that's why it looks as blue. So therefore here also we have a defect in one gene in a chromosome, but that leads to its manifestation in two different systems such as bone and in the eyes. So therefore if there is defect in one gene and there is manifestation in multiple systems, then it is called as pleiotropy. The other concept we are going to discuss now is variable expressivity. So we know that in different people with autosomal diseases, different people may present or express the disease in different ways with different severity and different manifestation. For example, if we have 10 patients who have suffer with Marfan syndrome, all of them will not have the same severity, all of them will have not have the what we call, all of them won't have the same age of onset or the same type of manifestation of Marfan syndrome. For example, some of the people will have all the problems of all the systems such as skeletal problems, ocular problems, and cardiac problems, and maybe their age of onset is 25 years old, whereas other set of people might have only skeletal and cardiac problems and their age of onset can be 35, whereas another group of set may have only ocular problems. So this means that different people with the same disease can have the different severity and different age of onset. This means that many variations are possible. So different Marfan's suffering people may have combination of clinical manifestation, and this is termed as variable expressivity. So basically it means that different people won't be expressing the same disease in the same manner. It will be varying from person to person, and that is called as variable expressivity. Now we are going to discuss the concept of new mutation. So new mutation is the type of mutation which occurs in the gametes of either the father or the mother. So this means that there was the copy, the mutant copy was present in the zygote, which means that the zygote got the mutant copy from either the sperm or the ovum, but the father and the mother, their genotype is normal. So for example, here we'll look at a pedigree chart and we'll consider the Marfan syndrome to occur all of a sudden. So here the grandparents are normal, all the parents and uncles are normal. So father was normal, the mother was normal. But when they gave birth to a daughter, the daughter had Marfan syndrome. So how come this happened? This means that either the mother's ovum or the father's sperm underwent mutation, and one of that was found in the resultive zygote, due to which there was the presence of Marfan syndrome. What is the risk for other children that they will also have new mutation, or what we call they will also be defective, born defective? So actually the risk is very low because the mutation, because if it was a new mutation, the new mutation occurred in one sperm out of millions of sperms. And since only that sperm contributed to form the zygote, other sperms are mostly healthy. So the risk of what we call developing the disease in future children is going to be low. Mutation in germ cell can take place in either of one of the parents, either in the ova or in the sperm. And then after that, if this Marfan syndrome woman marries someone, then the risk of her kids developing the disease is going to be 50% because it is autosomal dominant disease. The risk factor of another type of new mutational disease, which is very common, is the Down syndrome disease. That is, for example, if Down syndrome is not present within the family at any stage, but all of a sudden appears in the new offspring, it means that there must have been new mutation which took place. And the greatest risk factor of Down syndrome is increase in maternal age as well as increase in paternal age increases the risk of new mutation and especially the development of Down syndrome disease. Another disease which is an example of new mutation is achondroplasia. In achondroplasia, the receptor for fibroblast growth factor receptor number 3 is mutant. Due to this, the resulted baby will have short limbs, that is, he will have short arms and short legs, but his skull or face and trunk, that is chest size of the bones, will be normal. Why is this so? In achondroplasia, as we know, the fibroblast growth factor receptor 3 is mutant, and also we know for growth of limb the cartilaginous plate is present in the long bones, and in those long bones where the cartilaginous plate is present, they have these fibroblast growth factor receptor 3, and if they are mutant, these cartilaginous plate will fuse early in life, okay, and the limb will not grow fully. As a result, the baby will grow with short limbs. It is not dwarfism because the full body is not small, only the limbs are small, whereas the face and the trunk are of normal size. There is another concept that we have to get clear with. So, for example, in a particular city, if there are 1,000 achondroplasia patients and most of them are not reproductively fit due to maybe any different reasons, okay? So this means that generation after generation, the disease should become less prevalent. For example, here in our case, achondroplasia, it should become less prevalent because the autosomal dominant disease suffering people are not that much reproductively fit, and they are due to different reasons. Okay, they are not producing much kids. So basically, the disease should decrease. But scientists found out that this is not the case. Even though the patients who are suffering with achondroplasia are not reproducing much, but still the prevalence of achondroplasia instead of decreasing is pretty much high in the society. The scientists found out that there is high frequency of new mutations, and also in achondroplasia only 20% of achondroplasia patients have familial history, whereas 80% of the people who suffer from achondroplasia are new mutations. So there's a funny joke that marrying older men will bring you money, but it will also bring you small limbed baby. This is because the new mutation rate is very high, especially in children who are fathered by old men. So now we have understood the concept of different types of autosomal dominant pedigree charts. We have understood what is pleiotropy. We have understood what is variable expressivity. We have understood what is reduced penetrance, and we have also seen what is the concept of new mutation. So in now let us do a recap of the autosomal dominant and all the things I mentioned. Just let's clarify some more points about their concept, like such as pleiotropy, penetrance, and new mutation and expressive and sorry variable expressivity. So in most of the autosomal dominant cases, we are going to have a homozygous parent of normal allele, homozygous normal allele parent, and a heterozygous parent who is going to have the what we call abnormal gene, dominant gene. Okay, so then we have the following offspring, out of which 50% of the offspring will have a chance of having the disease, 50% of offspring will have the chance of not having the disease genotypically. The important point is genotypically there is a 50% of chance that they are going to carry the gene and 50% chance genotypically that they are not going to carry the disease. But if in any case the offspring who is carrying the defective gene does not express the phenotypic manifestation of the autosomal dominant disease, then we don't call that such a person carrier, but we are going to tell such a person that he has reduced penetrance. Because in autosomal dominant disease, usually one defective allele is enough to produce the disease. But sometimes in autosomal dominant disease, if defective allele is not producing the abnormal phenotype, this type of situation is said to be as reduced penetrance. But carrier word is used when we are talking about autosomal recessive conditions or even sex linked recessive condition. Usually, most of the people in autosomal recessive disease or people who are in autosomal recessive cases who are having just one defective copy have normal phenotype, but people with one defective gene of autosomal dominant disease usually have the abnormal phenotype. In many very rare cases only there is reduced penetrance. So therefore, for autosomal recessive, if a person does not express the autosomal recessive disease, we call him as a carrier. Whereas in autosomal dominant disease, if the person does not express the disease, we call him as reduced penetrance. So this sums up our concept that in autosomal dominant disease, when we speak that one homozygous normal parent is going to mate with a heterozygous affected parent or non-affected parent with reduced penetrance, the probability of the what we call progeny to have the defective genotype is going to be 50% defective genotype and 50% normal genotype. But these people with abnormal genotype will have abnormal phenotype or normal phenotype depends on the phenomenon of penetrance. So 50% chance of defective genotype being present is possible, and 50% of normal genotype being present is possible in autosomal dominant condition between homozygous parents and heterozygous parent. But the what we call presence of phenotypic expression of disease in those individuals who have the genotypic abnormality depends on the penetrance. Probability of the abnormal genotype to convert into abnormal phenotype depends on penetrance. Reduced penetrance means more people with abnormal genotype but they don't have the abnormal phenotype. That means they have the abnormal genotype but they have the normal phenotype. So therefore penetrance is directly proportional to phenotype. Penetrance is all or none, that is either it is manifested or not manifested. Now we are going to observe this pedigree chart to explain one more thing, and that is going to be related to variable expressivity. So here we had the homozygous parent and the heterozygous affected parent, and so we had the kids, for example 1 2 3 4 5 6 kids we had. Okay, out of them 4 kids have the defective genotype, and out of those 3 of them expressed also defective phenotype, but the fourth kid who had the defective genotype did not express the defective phenotype. Therefore in him, what we call, since it's a boy, there is reduced penetrance. Now when we talk about variable expressivity, we are going to talk about only those people who have an abnormal genotype as well as abnormal phenotype. We will not consider this person when we talk about variable expressivity. Why? Because in this person he has the abnormal genotype but he does not have the abnormal phenotype, so he does not have the disease and there is no manifestation of disease. Whereas variable expressivity, we remember, it means that different people with autosomal dominant disease may express the disease with different severity and different manifestation and at different age of onset. For example, if we consider that this parent had Marfan syndrome and all 3 of them suffer from Marfan syndrome, whereas this person carries the gene for Marfan syndrome but does not suffer due to reduced penetrance. So now variable expressivity means this person may only suffer from ocular and cardiac problem, this person suffers from cardiac as well as skeletal problem, and this person suffers with all the 3 problems of ocular, skeletal and cardiac. So we can say that we can compare variable expressivity in them but not in this individual because he does not suffer from the disease. So this means that in people who are suffering with reduced penetrance or who are blessed with reduced penetrance, we cannot apply the concept of variable expressivity to that person. Whereas variable expressibility should be applied to only those individuals who are affected with the disease and have clinical manifestation, and this means that in them the penetrance is 100%. So till now we considered the pedigree of a homozygous normal allele parent and a heterozygous parent. Okay. Now what if we consider the cross between two heterozygous parents? Okay. What is their progeny going to look like? So in their progeny, the 25% of the progeny is going to have normal genotype as well as normal phenotype. 50% of the genotype, or we can say 75%, okay, 75% of the offspring is going to have abnormal genotype, out of which 50% of them are heterozygous of mutant allele, whereas 25% are homozygous of mutant allele. So this means that if we cross two heterozygous parents, then we are in autosomal dominant disease, we are going to get 25% chance of the kid having normal genotype and normal phenotype, whereas 75% chance is going to be abnormal genotype, and out of that also 50% are going to have heterozygous and 25% are going to have homozygous mutant allele. And out of these progeny, these 50% of progeny with heterozygous are going to have less severe manifestation of the autosomal disease. So if we consider Marfan syndrome, they will have less what we call less severe manifestation of the Marfan syndrome. Whereas in this kid who is homozygous of mutant allele, he is going to have very severe manifestation of Marfan syndrome. And such kids who are having homozygous of mutant allele, when two heterozygous parents cross with each other, usually they are aborted or they do not reach the reproductive age and die early. So we came to know from this that most of the autosomal dominant cases are due to cross between homozygous parent and a heterozygous parent, or a heterozygous parent and a heterozygous parent. When we cross a homozygous parent and a heterozygous parent, the probability of the offspring having the abnormal genotype is 50% abnormal genotype and 50% normal genotype. Okay, here this is the offspring, this is the offspring, this is the offspring, this is the offspring, this is, so there is 50% chance of a normal offspring and 50% chance of abnormal offspring. Whereas when we cross two heterozygous parents, the probability of having normal genotypic and phenotypic what we call offspring is only 25%, whereas 75% of the progeny, there is chance for the progeny to have abnormal genotype, and out of that also 50% of the offspring are going to have heterozygous and 25% are going to have homozygous of mutant allele. Another important point to remember that in autosomal dominant pedigrees, most of the diseases are present in successive generation. So we will have a vertical pedigree. This means that here the grandparents had the disease, then their kids had the disease, and then their grandsons or granddaughters had the disease, and then their great granddaughters and grandsons will also have the disease. So the disease is moving vertically from up to down. That's why this is called as a vertical pedigree. So autosomal dominant pedigrees are vertical pedigree. In autosomal dominant diseases, male and female both can transmit the disease, and there is equal number of male and females affected, 50-50%. Now what if this couple who is heterozygous comes and asks us that we want to exactly know what is the probability of our kids having abnormal genotype, phenotype, etc. So we have three information. The first is the disease they have is autosomal dominant, and then we know that both the parents of heterozygous. And let us suppose that penetrance is 50%. So what can we say, that 25% of their kids, there is probability of the kid to have normal genotype and normal phenotype. 50% there is probability of having heterozygous genotype, and 25% of having homozygous of mutant allele genotype. And in this 50%, 25% will have genotypically abnormality, but phenotypically they'll be normal. whereas there is chance of 25% having genotypically abnormal and also phenotypical manifestation. Whereas where there is 25% chance of homozygous mutant alleles, there is 100% chance of phenotypic expression as well 100% genotypic expression of the disease. So now if they ask about the another child, what is the probability? We are going to say that 25% chance of having the normal kid, 50% chance of having abnormal genotype, but out of that 25% chance of having normal phenotype and 25% chance of having abnormal phenotype, whereas 25% chance of having a very dangerous combination which is going to die early or can also be aborted. Now we are going to discuss about autosomal recessive disorders. So a question is that how many of us are having at least one defective allele of some kind of autosomal recessive disease on our autosomes? So the answer is 100% of us are carrying 1 to 5 mutant allele for autosomal recessives, and we all are carriers of 1 to 5 autosomal recessive disorders. Now, for the better understanding of autosomal recessive diseases, we are going to understand it based on this pedigree chart. So we are going to take example of a South African white family. So South African white family during the apartheid time used to just marry within themselves and did not marry any black people who used to live in South Africa. Due to this, there was a disease which was running among them, and that was called as Porphyria variegata. So in the starting, it is like long ago, both the white people who started the disease were normal. But what happened? Either the male or the female, in one of their gametes, for now we'll consider the female, her ova underwent mutation due to some, no, I don't know what we can say, some radiation or some other cause, and eventually when she had the offspring, the one of her offspring was the carrier. So generation after generation, the carrier gene was passed on to further what we call generations. But the disease Porphyria variegata was not expressed until the defective gene found its better half or another defective gene to form a homozygous, what we call individual genotypically. So as we can see here, we had genotypically eventually carrier, and so eventually this lineage of family, this when there was a current type of a cousin marriage, we had 4 offsprings, out of which the probability was that 25% were normal genotype and phenotype, and 25% was the place where the disease was manifested because it was a homozygous of mutant allele, and that is what is required for autosomal recessive disease, whereas 50% were carriers. So, if the South African white people kept marrying among themselves, eventually they will end up with more cases of Porphyria variegata. So, this means that if we cross with each other, there are more chances that the mutant allele will meet the other mutant allele and the disease will be manifested. So, now here also we can see further carriers present in the generation after generation, but the disease will only be experienced when two mutant alleles are expressed together. There is homozygous mutant allele in the genotype of the progeny. Now, if we imagine that some of these individuals from this generation migrated to Germany and they are the carriers of Porphyria variegata, whereas in Germany, the individuals whom they are going to marry are the carrier for the hemophilia A. So, this means that when this South African white family descendant and German family descendant will cross with each other. The African family had the Porphyria variegata defective gene and the Germany family had the allele for hemophilia A. So the baby which are going to be produced may have one defective copy of both. That is, they will have the defective copy of the gene for Porphyria variegata as well as the defective copy of the gene for hemophilia. But the babies will not develop the disease. This is because to express or manifest the disease, the progeny or the baby should have two recessive alleles. But since they have one recessive allele for one disease and one recessive allele for another disease, they are not going to express any of those disease. So this sums up that distant marrying is good if seen on a long-term scale. This is because in distant marriage, the defective allele don't become homozygous. Most of the autosomal recessive disease are produced due to close marriages, which is called as consanguinity. The chance of autosomal recessive disease increases if there are close marriages in isolated people, in religious people, example Ashkenazi Jews who will marry among themselves only, and among tribal people because they are located at a very high altitude and they don't want to go to different places to get married. So as a result, in such people, mutant alleles will keep meeting with each other and result in homozygous individuals, and the disease will be manifested. Not only this, autosomal recessive, when mutation occurs, it takes many generations until the disease is manifested, but autosomal dominant conditions will be manifested immediately after the new mutation takes place. In autosomal recessive disorders, the gene should travel many generations to manifest itself. As we saw here, the mutant gene started from this generation, but it had to go for 1, 2, 3, 4, 5 generations. After 5 generations, only the first, what we call, clinical symptoms of the disease was manifest in the kid. So this means that autosomal recessive disorder, the gene should travel many generations to manifest, but it is more common when there is consanguineous close mating. Whereas in autosomal dominant, only one allele is enough and can produce the disease. Not only this, the pedigree in autosomal recessive is horizontal, whereas the pedigree in autosomal dominant is vertical. Horizontal means that here there was no disease, here there was no disease, but the disease was here and most of them were carriers horizontally. So this is what is called as horizontal pedigree, and it is for autosomal recessive. Autosomal dominant and autosomal recessive disease both affect male and female in equal number. Here one important point to remember is that autosomes also determine the sex. That is, X and Y chromosome determine the sex, but autosomes play an important role because many of the hormones which are produced, for example FSH and LH, their genes, that is the genes of FSH and LH, are present on autosomes. Example, if the zygote will have XX genes, then those genes are activated, on the autosome, which will produce female phenotypes. And if it is XY, what we call genotype of the zygote, then those genes are activated on autosomes, which will produce male phenotype. X and Y are sex determining genes, but sex associated genes may be present on autosomes. For example, testis produces testosterone, but to make testosterone, the enzymes which are required are synthesized by genes which are present on autosome. But the activation of these genes or their inactivation depends whether it is a female or a male. So if you are a male and have XY, then those enzymes which are responsible for production of testosterone will be activated, whereas if you are a female XX, then those genes which are responsible for production of testosterone will be inactivated. We're going to discuss about locus heterogeneity. So what is locus heterogeneity? Locus heterogeneity states that sometimes mutant alleles are present at different loci at different chromosome, but they produce the same phenotype. For example, we will consider this as John's family and this is Robert's family. They are going to produce an autosomal recessive condition which is associated with ears and causes deafness. So the ear is made up of middle ear and inner ears. If we think that homologous chromosome 1 are responsible for formation of middle ear and homologous chromosome 7 are responsible for inner ear, if there is defective genes which are present on both the parents in John's family, as he is homozygous or mutant allele, he won't have the middle ear and he will be deaf. Whereas in Robert's family also deafness is present, but the problem here is with inner ear because On seventh chromosome, the genes which are defective alleles of each other are responsible for formation of inner ear. As a result, if those alleles of the genes formation for inner ear defective, even Robert's family, Robert won't be able to hear. He will have deafness. So here we saw mutant alleles at different loci, at different chromosome, but they produce the same phenotype. The same phenotype is deafness, and the alleles are here, they're at this loci, here they're at this loci, and they're at different chromosome 1 and 7. This means that many people will have deafness, deafness, and that may be due to underlying different genetic loci with different mutation, different chromosome. Like for example, John's and Roberts people develop deafness, but the reasons are different. If John's family marries with each other more and more, deaf people will be born in their family. Whereas same goes for Robert's family. If they will also be marrying more among themselves, more deafs will be born in their family. But what happens, we will see in their pedigrees. For example, if we take this as John's pedigree, so we saw 1 chromosome and 7 is present, for example. So one defective from here, one defective, heterozygous parents. So 25% of the kids in John's family, there's a responsibility possibility that they will be normal, 50% will be carrier, and 25% will be the deaf kids born. Same goes if we see in Robert's family, heterozygous and 25% are deaf, 50% are carriers, and 25% totally normal. Now what happens if we marry a deaf from John's family and a deaf from Robert's family? So if we take a deaf from John's family and deaf from Robert's family and marry them, the result will be that all the kids will be normal and they won't be deaf because they all will be carriers. This is because deafness is produced in different people due to genetic defect at different loci, and this is now we are going to discuss about sex linked disorders and sex limited disorders. So first let us see what are sex limited disorder. So sex limited disorder means those disorders which are limited to one gender, either male or female, and are caused due to the genes which are present on the autosomes. Example, male pattern baldness. The male pattern baldness disorder is present on both male and female genes, but it is expressed only on in males due to high testosterone level in males as compared to females. Another example is ovarian cancer. It may be inherited due to autosome present in both males and females, but males don't have ovaries, and so the mutant allele cannot produce ovarian cancer in males, but of course it can produce the ovarian cancer in females. Now we will discuss sex linked disorders. So we have two types of sex linked disorders: X linked sex disorders and Y linked sex disorders. So we should know that X chromosome is big as compared to Y chromosome. Y chromosome is small, and very few genes are present on the Y chromosome. Y linked diseases are not common. Y is given from father to only the son, and it will never go to daughter. So as a result, Y linked diseases, if there is, are only transmitted from father to the son. So if we see the Y-linked pedigree, only after the father, the sons will be affected and the grandsons will be affected. One example is people who are having ears with hair or have hair on their ears are said to have Y-linked disorders. There are very few genes on Y. Genes on Y will basically determine the capability of the male to reproduce. So if the Y gene has some kind of mutation or some kind of defect which can cause Y disorder, it will affect the reproductive capability of the individual or the male. Now we are going to discuss about X-linked disorders. So in X-linked disorders, we should know that in females we have two X, and one of X is from paternal X and one is maternal X. So at blastocyst stage, the females will inactivate randomly in some cells their paternal X chromosome, and in some cells they will randomly inactivate their maternal X chromosome. This happens at random at blastocyst stage. So inactivated X chromosome is called as bar body. So the females will have one bar body in each cell. And females are also called as genetically mosaic because if a person in his body, in his somatic cells, are derived from more than one genetic combination, then that person is called genetically mosaic, because in some cells which are somatic cells derived in females are from the paternal X chromosome, which is from a different line of cells, whereas some of the cells are derived from maternal X chromosome, which is another line of cells. That is why females are genetically mosaic. So in a normal female who has 2x, there will be one bar body. In a female with 3x, there will be two bar bodies. And in female who is suffering from Turner syndrome, there will be no bar body. So we know that XX, maternal X and paternal X, and in males we have XY, maternal X, and Kanyashna, paternal Y. And so in males, the one X which is present will always be active, because for normal functioning of the body, one X chromosome should be active. In males, that one will be active, whereas in females, sometime in some cells maternal will be active, and in some cells paternal X chromosome will be active. Now we need to remember that males will give their Y chromosome to sons and X chromosome to daughter. So if there is a X-linked disease present in males, then it can only be transmitted to daughters. Male cannot transmit the X-linked disorder to son. Whereas women can give either one of the X to male or female. So women can transmit the X-linked disorder to males as well as females. X-linked disorders can be divided into X-recessive and X-linked dominant disorders. Most of the genes present on X chromosome are enzymes related, so most of the X-linked disorder are X-linked recessive disorder. Very few are dominant disorders. Now we are going to study more about the X-linked disorders. First, we are going to discuss the X-linked recessive disorders. So here, if we consider a heterozygous female who is a carrier of a disease, if she marries a normal man, what is the progeny going to look like? And we should remember that in females, sometimes, as we know, in some cells paternal X is active, and in some cells maternal X is active. So in some cells of this female, the mutant X will be active, in some cells the normal X will be active, but that is not going to affect the female in much great scale. She might have some molecular changes but not much, and still she will just act as a carrier. So we are going to get carrier female, normal female, carrier affected male, and normal male. So this means 50% of the daughters will be carriers and 50% of the males will be carriers. But in the case of males, we cannot say them as carriers. We call them as hemizygous. Why? In males, there's just one X chromosome, and if the mutant X chromosome is present, then that X will be what we call expressed. So this means that because only one X is present, and that only X mutant allele for an enzyme. Okay, so if this X is mutant allele for an enzyme, okay, for its responsible for production of an enzyme, then that enzyme won't be produced, and there is not another X to produce the enzyme, because in female we have two X. So maybe if one X is mutant, the other X might produce the enzyme, but in males that's not the case. That's why they're called as hemizygous. So even though recessive in presence of a healthy allele, it will not manifest. Okay, so since this is a recessive gene, and if it were had another X healthy gene, it won't manifest. But in the case of male, we there is not healthy allele, there is just one X chromosome, and that here is mutant. So truly speaking, in case of males, we cannot say X linked recessive or X linked dominant. This concept does not apply to male, because in both cases the disease is manifested. As in males, there is just one X chromosome, and we know to live a healthy life at least one X chromosome should be expressed. So what if your luck is so bad, you get the mutant X, that X chromosome is going to be expressed, and as a result you are going to have manifestation of the disease. And so due to these reasons, we truly say that X linked recessive or X linked dominant concept does not apply to males. Second case is that if this affected male marries a normal woman, what is the outcome? So if this affected male marries a normal woman, the male can give the X chromosome to the daughters, so both the daughters are going to be carrier. But the male cannot give the X to his sons, he only gives the Y. So both the sons will be normal. As a result, when we have an affected male and a normal female we will have 50%, what we call 100% of the female daughters will be carriers, whereas the sons will be normal. Now, if we cross an affected male and a carrier female, what will we get? So we will get an affected male, a normal male, and an affected woman and a carrier woman. This is a very rare scenario where we have an affected woman. It is unusual situation in which a female is affected by X-linked recessive disorder. So in this pedigree, what we can see is that 25% of the males are affected, 25% males are normal, 25% of females are also affected, and 25% of females are carrier. Now, how to identify the pedigree charts? Okay, so if any pedigree chart, first we have to understand whether it is autosomal pedigree chart or it is an X-linked pedigree chart. So in autosomal pedigree charts, the affected males and females will be equal. That's the main clue. And in X-linked charts, pedigree charts, the male and female affected will be imbalanced. Now X-linked and autosomal. So if there is no father to son transmission, we have to understand this is X-linked transmission, whereas in autosomal transmission there is father to son transmission as well as mother to son transmission, and male is to female ratio is equal, that is equal number of males and females are affected, whereas in X-linked unequal number of males and females are affected and there is no father to son transmission. And if you see vertical transmission, then it will be autosomal dominant, and if you see horizontal transmission, then it will be autosomal recessive pedigree chart. Now in X-linked, as I told, there is no father to son transmission and the ratio of male and female affected will be unequal. If more females are affected than males, then it is X-linked dominant disorder pedigree chart, and if it more males are affected as compared to the female, then it is X-linked recessive pedigree chart. This is because males have only one X chromosome, and if that X chromosome by mistake is the mutant X chromosome, then they are surely going to suffer. Sometimes it happens the carrier females of X-linked recessive disorder, for example, like we saw in this cross, they can also be effective. How? We know that the females have two X chromosome. So sometimes if by mistake the females, we should know in their body some 50% of the cells should have paternal X chromosome functional and some 50% of them should have the paternal X chromosome, maternal X chromosome function. Now sometimes what happen if a female has a mutant X chromosome, the carrier female, okay, and what she does is by mistake her body activates 70% of the mutant X chromosome and 30% is only active is the healthy X chromosome in the 30% of cells, and in remaining 70% of cells the mutant X chromosome is active. As a result, there will be manifestation of the X-linked recessive disorder in such female. Now we are going to discuss about X-linked dominant disease. So example of X-linked dominant disease is hypophosphatemia rickets. So in these patients, from proximal renal tubule, reabsorption of phosphate is poor. As a result, a lot of phosphate is lost in urine, which results in hyperphosphaturia. As a result, when the level of phosphate is low in blood, we cannot mineralize our bones properly. To mineralize bones, we need to have specific ratio of calcium and phosphate. If the ratio of calcium and phosphate is not perfect, the bone will be weak due to poor mineralization, and the bones will be able to bend too much, and this is called as rickets. The protein recapturing phosphate, its gene is present on X chromosome. So the protein which is present in proximal tubule is what we call having its gene present on the X chromosome. So in these phosphate regulatory proteins which are present on X chromosome, even if one X chromosome is defective, this disease will be manifested. Now we are going to see the pedigree of this X-linked dominant disorder, that is hypophosphatemia ricket. So if we have an affected female and she mates with a normal male, we will have a normal female, affected female, a normal male and affected male. So there is 50% of progeny will be affected. Now if we cross this affected female with a normal male, in her generation, in her progeny, only one female will be affected and one male will be affected. So again, there is 50-50% chance. But now if we cross the affected male with a normal female, both the daughters of this male are going to be affected, and both the males or the sons of this male are going to be not affected. So there is no father to son transmission we can see here. And also the manifestation of the disease is more severe in males as compared to females, because in males there is just one X chromosome, and if that is the mutant X chromosome, it is going to manifest the disease in very severe, much severe manner. Whereas in females also it's going to manifest the disease, but it's going to be slightly less because the female has two types of X. And also in females we have the one X is enough to produce the disease, but the disease can have two level of suffering, mild and severe. In mild there will be hypophosphatemia but without rickets. That means phosphate is being lost by the kidneys but not at a rate, not at very high rate, so that some phosphate is left behind and that can help in the mineralization of the bone. Whereas in the severe case, a lot of phosphate is being lost in the urine, and so the bones cannot mineralize properly and they will be bendy, and that will cause rickets. In the starting, the male to female ratio was normal, but as the pedigree moves forward, the number of female sufferers becomes more than the males. Especially whenever affected male mates with the normal female, all the daughters affected and none of the males are affected. For example, here when the affected male mates with a normal female, all the daughters of his are going to be affected. So, and when the affected female mates with the normal male, male and female are equally affected. So, when an affected female mates with a normal male, equal number of males and females are going to be affected. So, X-linked dominant pedigree and X-linked recessive pedigree, there is no father to son transmission. In an X-linked dominant pedigree, females will more be affected, and in X-linked recessive pedigree, males will be more affected.